All right, everybody, continuing where we left off the last time in Chapter 6, talking about chromosomes and cell division. So we've been talking about how DNA and genes and chromosomes are passed along from parents to offspring. One of the things that you can note about all of the individuals in this picture is that they all look similar but not exactly alike. And that is because they are brothers. And so they all have similar genes, similar DNA being passed from parent to offspring, but that does have a little bit of variety, and that's what makes them not identical. That's what makes them look a little bit different. And so as you have been studying in your lab, parents have the opportunity to pass along these alleles from parent to offspring, and one or the other of the alleles is being passed along, and so that's why we do have some variation we see in offspring. In talking a little bit about cells, we are going to talk about how we have different types of cell division. This picture is of bladder cancer cells, so these are cells that we don't want to be dividing. We start thinking, when we talk about passing down our traits from parents to offspring, that at some point our cells, after having divided and divided and divided so many times, that we get to the point where they're not capable of dividing anymore, and that point is when the cells stop dividing and we start to have cell death. And so if we have cells, however, that continue to divide forever, at some point the quality of those cells is going to deteriorate and can then pass on different types of degenerative disorders and basically cause sickness and cause death. So as we're growing, we're having some of the same cells are growing and dividing and then growing into their own cells and dividing again. You don't just have one set of cells that is you from the moment you're born until the moment that you die. Some cells are going to be continuously regenerated, and some cells, once they die, that's the end of them. There is no moving on from that. So the way that cells know whether or not they're going to divide is based on what's called a telomere. So every time a cell divides, the telomere gets shorter. You can see here we have our picture of our functional DNA, and it basically has a little cap on it, a protective cap at the end of each chromosome that says, okay, we're protecting the end of the chromosome, and as it divides and divides and divides, we see here it's getting shorter and shorter and shorter, and then eventually when it breaks off the tip of the chromosome, that's when the cell death occurs. And so at birth, telomeres in most human cells are long enough to support about 50 cell divisions. So most of your cells are going to be able to divide about 50 times before they die. So as we've been talking about cloning, and we've been talking about can your cells live forever, or this fountain of youth, we know that cells can only possibly divide about 50 times. And so if they continue dividing after that, then you're going to lose this functional part of the DNA. So the telomere is like a cap at the end of the DNA, and every time the cell divides, the telomere gets a bit shorter until eventually, when there is no telomere anymore, that's when we have cell death. And so in certain types of diseases like progeria, we see that the cells are unable to support these multiple cell divisions, so we have rapid aging occurring. This person who has progeria is actually a young child who looks like they are very, very old because their cells have lost those telomeres very, very quickly, so they take on an aging appearance and they tend to pass away when they're very young. Scientists who are looking at telomeres are trying to figure out how we can keep getting longer and longer telomeres so that we can have our cells survive longer and longer periods of time. So they might be able to figure that out at some point, and then your cells could continue to divide forever. If you go online and look up diseases like progeria, you can see it's a rare condition that happens when the symptoms are that a normal human appears to have aged very quickly, but it occurs in very young children, usually not passed on through families. So this would be considered a disorder that wouldn't be passed from parents to offspring. It would be seen very, very rarely, and it can be it is a very deadly disease. When we look at things like cancer, in cancer we have cells that rebuild their telomeres after each cell division, that restore that protective cap then and never stop dividing. So if we look back at this picture, if we have a cancer cell that is occurring in the body, this telomere keeps getting rebuilt every single time so that the cell keeps dividing and growing and dividing and growing and never stops dividing and growing. So cancer is marked by cells that divide and grow without ever having that mechanism to stop. And so as they continue to grow and divide, these cancerous cells steal the body's energy, make the person who has cancer very, very tired, and if they don't go through chemotherapy or radiation, then there's no way to stop these cells from growing, making tumors, and eventually killing the host. So basically what's been said about cancer cells is that they are those which have forgotten how to die. So because cells usually only divide 50 times and cancer cells continue to divide and grow forever, they are considered cells that have forgotten how to die. The way that these cells are going through this division and growth is through a process called mitosis. Mitosis has just one purpose, and that is to create new genetically identical cells. So the two reasons that we need our cells to grow and divide is that we need to, number one, grow. So as we're getting bigger and bigger, we need to have more cells. And then, so that once you reach your final stages of development and you're into the aging period, you want to replace the cells that have been destroyed or hurt in some way. So repairing skin cells, repairing any cells within the body that have been damaged, and then at some point when you're not growing and you're not replacing your cells anymore, that would be when you are dying. So in looking at mitosis, looking at the way we go from having one cell to having multiple cells, you can see the first stage in mitosis is where we have our nuclear membrane here that is around all of our chromosomes. Our chromosomes are going to begin to replicate. The centromeres are over here on the side. They're getting ready to pull the chromosomes to either side of the cell. And so interphase is basically this preparation for mitosis. One of the things that we've noticed about animal chromosomes is that they are linear. They look like the letter X in a picture because they have sister chromatids, which are one set of information on one side and exact duplicate of the information on the other side. They have a centromere that's holding them together. Sometimes they look like they're all squiggled up or they're all wound together, but they look like this X because it has a centromere holding it in place so that it can begin to divide. And in the sister chromatids, we have this single chromosome, and then through replication we have a exact duplicate of that chromosome that is then attached there in the middle through this centromere, and the chromosome and its identical replicated copy are joined at the centromere. Then in mitosis, you can see we start to have a breakdown of this nuclear membrane where the sister chromatids are getting ready to be pulled to opposite sides of the cells. So we have our prophase here, where the nuclear membrane breaks down. The sister chromatids are replicated, so we have two duplicate copies, and they are condensed together. And these spindles are starting to form, which are going to attach to these chromosomes and get ready to pull them apart. So then the next phase in mitosis is metaphase. Metaphase is where they line up here along the cell's equator. They are attached to these spindle fibers, which are attached to the at each end of the cell, getting ready to pull it apart. So we have them lined up in the center, getting ready to separate. Then as the chromatids are pulled apart by the spindle fibers, notice one exact duplicate goes to one side of the cell, one exact duplicate goes to the other side of the cell. And then in the telophase, they start to cleave down the middle, and they are getting ready to separate and become these two distinctly separate cells. Okay, so we have then cytokinesis, where each of the two daughter cells is completely separate from each other. The cytoplasm and organelles and everything are going to begin to duplicate here in each of the individual cells in equal parts. So notice we have two identical cells that are completely split from each other and are going to then go on to split again. So when we are looking at things like cancer and looking at diseases like cancer, we start to see things like tumor growth, where we have unregulated cell division. All of these other cells within the body know that they are going to grow and divide and grow and divide, and they're going to do that 50 times. But tumors don't know that. They are going to continue to grow and divide forever. So cancer cells have several features that distinguish them from normal cells. So here in these two petri dishes, we see that normal cells are just going to be growing and dividing, and when they bump up next to one another, they recognize cells next to them and are going to stop dividing. Cancer cells do not know that, however, and so whenever they bump up next to each other, they just continue to grow, piling on top of one another. So the name for this is called contact inhibition. Contact inhibition is when cells make contact with one another. They begin to stop dividing in normal cells, but when cells make contact with each other in cancerous cells, there's nothing there to inhibit them from growing. So here we see normal cells splits into 2, 2 splits into 4, 4 splits into 8, and eventually they no longer are able to divide. But in cancer cells, 1 splits into 2, 2 into 4, 4 into 8, and 8 continues to grow forever, but they start to become jagged cells that are full of things that don't work correctly, that grow and divide out of control, and that's why cancer is such a deadly disease. When we look at these pictures of the body, we can see that the cancer cells here are shown in bright yellow, and they can separate from a tumor and move all around through the body and through the circulatory system. So whereas we might have cancer starting in one small tumor, eventually as it breaks off and it starts to move around to different parts of the body or spread to different parts of the body, that's when treatment becomes so difficult, and that's when cancer becomes deadly, because killing off a tumor or a tumorous growth of cancer is something that is possible, but once it starts breaking off and moving around the body, that's when it becomes very dangerous, and that process is